AS Level Biology 9700, Paper 2, Variant 1, May June 2025, question number 3. Already we have discussed in video 1 the question number 1 and question number 2. Now this time we are going to discuss the question number 3. So question number 3 starts as scientists investigated the progress reactions catalyzed by two enzymes, DOPA oxidase and nuclease. The reactions catalyzed by these enzymes result in changes to the appearance of the action, to the reaction mixtures. The reactions are shown in figure 3.1, which is L-dopa, colorless solution. In the presence of DOPA oxidase enzyme, DOPA chrome is formed, which is orange-brown solution. Other reaction, casein plus water, which is white solution. In the presence of nuclease enzyme, the peptides, short chains of amino acids, colorless solutions. Colorless solution is formed from white solution. So the changes in the appearance of the reaction mixtures make it possible to follow the reactions using a colorimeter. So figure 3.2 shows the progress of the reaction catalyzed by DOPA oxidase as recorded from a colorimeter, and figure 3.3 shows the progress of the reaction catalyzed by nuclease as recorded from a colorimeter. So both of these reactions has been shown in the form of graph, which you can see over here in the next page. First of all, the graph 3.2, which has taken place in the presence of DOPA oxidase enzyme. So in the presence of DOPA oxidase, you can say the absorbance rate is 0 when the action starts, and then it is increases and reaches to the maximum at after 300 seconds. So it reaches to a maximum level, which is 0.90. So it means it starts very fast and then it ends slowly and move towards the other graph, which is 3.3, in which the nuclease enzyme has been used instead of. So here you can see absorbance rate is decreasing. So it was highest, that is around 1.15, 16, 17, and then it is start reducing, and after 120 seconds it reaches to its end, but it at 100 and 110 it was equal. At 110 and 120 it was equal rate of absorption, which was at 0.10. So it means the reaction started very fastly and then it ended. Now the question is, with reference to figure 3.1, figure 3.2, and figure 3.3, describe and explain the similarities between the progress of the two reactions. So there are so many similarities which can be discussed over here. First of all, the first you can see the resemblance or the similarity between these two graphs is that is the both reactions change over time. The reaction progress to है that is changed after time. As for example, both reactions show a change in absorbance over time, indicating a chemical process is occurring. So I would write start that is the both reactions. Both reactions show a change in absorbance. Show a change in absorbance over time. indicates a chemical process. A chemical process is taking place. Is taking place. So this is the similarity in it. As the initial rate is similar in both of these, both reactions will have an initial rate of change. Whether it is increasing or decreasing. So, which is fastest, which is the fastest. Whether, again, I explain whether it is increasing or decreasing. In one case, it is increasing. In one case, it is decreases. That is difference. But we are talking about the similarity. Similarity is this, that both of they have the fastest reaction, fastest initial reaction. That is it is taking place very fastly because because the reactant concentrations are highest. Reactant concentrations are highest in the start. And then uh, secondly, for both type of reactions, the rate of change slows down as the reaction proceeds. As time passes, the rate of reaction for both slows down because the reactants concentration decreases. And then both reactions eventually tend to level off. At the end, both reactions eventually tend to level off. Level off and reaches to a point where where the no change in the adsorption. means to a state of equilibrium. So these reactions depend on many factors, but you can see these are the similarities between two types of reactions which are taking place. One, it is taking place in the presence of dopa oxidase enzyme, uh, and the other which takes place in the presence of nuclease enzymes. In both of these graphs, you can observe both of these curves which has been obtained from these two graphs, the similarities and differences as well. But they have asked about the similarities only. So similarity is means that both reactions show a change in the absorption. In one, it is decreasing, one it is increasing. But we have to show that in both of these reactions, the rate of reaction in the start is very fast, and then it is slow down, and at the end, it becomes there is a state of equilibrium when there is no change, there is no collision, there is no reaction taking place, and they because the reactants has been used up during these reactions. So that is the reason the reactions level off and they reach to a state of equilibrium. Next question is, suggest two advantages of using a calorimeter to investigate the progress of reactions such as those shown in figure 3.1. As we have talked about, that is these two reactions which were taking place in the start, which they have given in picture figure 3.1. So we are talking about that they were investigated in the presence of calorimeter. So I would like to discuss the role of this calorimeter or the advantages of using this calorimeter. So if we talk about this, just they are asking only two advantages. So first advantage can be that the calorimeter allows for the measurement of absorption of a solution without needing to remove samples from the reaction vessels. So this means the reaction can be monitored continuously in real time without disturbing its equilibrium or altering reactant concentrations by sampling. So traditional method might require stopping the reaction or taking aliquots, which can introduce errors or interrupt the natural progress. Secondly, high sensitivity and quantitative measurement can be taken with the help of this calorimeter. So calorimeters are highly sensitive and can detect even small changes in the concentration of a colored substance. So absorbance is directly proportional to concentration. So in this way, it provides a quantitative and accurate measurement of how much reactant is being consumed or how much reactant product is being formed. So this allows for precise determination of reaction rates and the point of completion equilibrium, even for reactions that proceed only in small amounts of colored species or where concentration changes as subtle. So I would like to write all these 2 points which I have told you about the calorimeter. So first is that is the calorimeter uh, allow the measurement of absorbance of a solution without needing to remove samples. So the reaction can be monitored by using reaction can be monitored continuously continuously in real time without disturbing reactant concentrations by sampling. So there is no need to take sampling. So traditional methods might require stopping the reaction or taking samples, which can introduce errors or interrupt the natural. So no introduction of errors. And second, it gives, it provides a quantitative and accurate measure of how much reactant is being consumed or how much product is being formed. So it allows for precise determination of reaction rates and the point of completion. So only for reactions, they produce only small amounts of colored species or where concentration changes are subtle. So this allows for precise determination of reaction rates and the point of equilibrium. So in this way, it is very helpful, and by using this, we have great advantages to perform the different enzyme catalase reactions. Now moving towards the next question is, scientists searching for a suitable enzyme to use in an industrial process isolated the bacterium Vibrio Farmia mollificus from the mouth of the Mediterranean eel Morina helena. The scientists discovered an enzyme in the bacterium that was suitable for the industrial process. The scientists named the enzyme VPSP37. The scientists investigated how the rate of reaction catalyzed by VPSP37 is affected by the concentration of substrate. The results of the investigation are shown in figure 3.7, 3.4. So 3.4, you can see the rate of reaction has been given on y-axis, and on x-axis they have given the concentration of substrate. Concentration of substrate. So the rate of reaction is very fast, and then uh, after certain it reaches to a an equilibrium. You can see straight line. So by keeping in mind this graph, calculate the Michaelis-Menten constant Km for the enzyme VP-SP37 using the information in figure 3.4. Show your working. We have to find out Michaelis-Menten constant from this graph Km value, and you know uh, this Michaelis-Menten constant is a measure of an enzyme's affinity for its substrate. Michaelis-Menten constant is a measure of an enzyme affinity for its substrate. So a low Km value indicates a high affinity of the enzyme for its substrate, meaning the enzyme can achieve half its maximum at a low substrate concentration. And a high Km indicates a low affinity, meaning a higher substrate concentration is needed to reach half V maximum. Km is numerically defined as the substrate concentration at which the reaction rate is half. Let me do it. Km is defined as the substrate concentration at which the reaction rate is half of the maximum reaction rate. So how can we calculate the Michaelis-Menten constant? First of all, we will go with the V maximum, and V maximum in this case is, let me use the smart pen. You can see the maximum. This is the I need a straight line. So this is the V maximum. And we can find out the Michaelis-Menten constant, half of this V maximum, by calculating the half of this V maximum. So half of this V maximum is, again, there is not a straight line. Let me do it again. As students, make use of scale, because I don't have, I am unable to use the scale on this laptop screen, which is touch screen. So I am not, it is not possible for me. I'm making use of this smart pen from, so this is half V maximum. Now the find out Michaelis-Menten constant, we will go the point where this line of half maximum strike with this. So we will go in the downward direction. We will draw this line in the down. So please again draw with draw with scale. So here will be 0.02. So it will be 0. It will be equal to 0.1 and the 0.01. So it is 0.14 or 0.17. So I would like to go with it. It will be nearly equal to 0.01. 0.014. So in this way, I would like to write over here, V maximum is 80, half V maximum is 40, and then if we at the rate of maximum, we from the graph the rate of 40 substrate concentration at half V maximum is 0.014. So this is the Km value from this graph. Moving towards the next question. The scientists discovered other enzymes that were suitable for the industrial process. These enzymes had higher Km values than VPSP37. Explain the advantage of using the enzyme VPSP37 in the industrial process rather than one of these other enzymes with higher Km values. So advantage of using this VP37. So I would like to discuss this question. As we have find out the VP SP37, why we are using this enzyme. So we need to recall that the meaning of Km that we just discussed in the last question. So Km is a measure of an enzyme's affinity for its substrate. Let me write it again for your convenience. Km is a measure of an enzyme's affinity for its substrate. So a lower Km value means higher affinity. A higher Km, low affinity. Means if the reaction is taking place very fastly and Km value is very low and the reaction has been proceeded in a short time, it means it has higher affinity. And if it is taking place very slowly, it means it has very low affinity. So it is taking time. So here this enzyme is very suitable because it has higher Km value, uh, VPSP3 से advantage of why, and then we are using VPSP3. So higher efficiency at lower substrate concentration. The first answer for this is it has it has a higher affinity for its substrate and low Km. So industrial in an industrial process, this is a significant advantage because the reaction can proceed efficiently and at a good rate, even when the substrate is scarce. Second point can be uh, this translates to cost savings. Savings and potentially faster reaction. Reaction times in situations where substrate availability might be limited. It means it has significant advantage. Reaction can proceed very efficiently and fastly. And then in the presence of very small quantity of a substrate. Even the substrate is in low concentration, the reaction can proceed very well. And so in this way, if it is taking place in the presence of very small quantity of substrate or very low concentration of substrate, it means it is cost effective. So because the substrate is, if the substrate is expensive, so it also give the cost effective measurement, so that so it can give a very advantageous, it plays a very advantageous role in you can say industrial processes. So they are making use of this type of enzyme. So it was all about question number 3. Please like, share, and subscribe my channel to watch the videos which are on the way. Thanks for watching.